Hello, and welcome to our webinar for Single Cell CNV, Achieving Single Cell Resolution of Genomic Heterogeneity. I'm Shauna, the 10x Community Manager and your moderator for today. Before we get started, let's go over some quick housekeeping items. Our speakers I'll introduce in a minute will be giving a presentation followed by an open Q&A session. We're recording this session and will make the recording available on the 10x website shortly. Please note that all attendees are on mute, and if you'd like to ask a question, please select the Q&A option on the bottom of your screen. Selecting the Q&A option will open a Q&A box so you can type your question. Please note that you can ask your question anonymously by checking the Send Anonymously option. And finally, if you're interested in tweeting during the session, please use the hashtag Celebrate. Okay, now let's meet your speakers for today. Up first is Anushka Brownlee, the Software Product Manager here at 10x. Anushka received her undergraduate degree in computer science from Carnegie Mellon University and her master's in bioinformatics from Georgia Institute of Technology. She previously worked as a life sciences IT and informatics consultant, and before that, a bioinformatician at Complete Genomics and the J. Craig Venter Institute. Joining Anushka will be Shimoni Maheshwari. Shimoni holds a PhD in genetics and is an application scientist at 10x. During her PhD and postdoc, she worked extensively in identifying drivers of genome evolution in both model animal and plant species. She now explores 10x data to answer interesting biological questions and discover exciting new applications of the technology. And now I'm going to hand it over to Anushka so she can get started with the presentation.
Thank you, Shauna. Welcome everyone to this 10x webinar, and thanks for taking the time to join us. Today we will be talking about our most recently released product, the Single Cell CNV Solution. The agenda for today is as follows. We'll start with an overview of the 10x products. Then we'll talk about the Single Cell CNV Solution, including an overview of what it is, details about the workflow, and what type of data is generated. I will then hand it over to Shimoni to talk about what you can do with the data. Shimoni will describe an experiment where we generated single-cell CNV data for over 10,000 cells to analyze spatial characteristics of genomic heterogeneity in breast tumor. There will be plenty of time for Q&A at the end of the webinar, so please don't hesitate to ask questions. To date, we have 4 product lines. All of our products are based on our Chromium system, which provides a precisely engineered reagent delivery method that enables single molecule and single cell microreactions at scale. In 2015, we released our linked-read genome sequencing solutions, which provide the benefit of having long-range genomic information to create better genome and exome assemblies, particularly across difficult regions of the genome. With this long-range genomic information, we are able to perform accurate variant calling, including identification of SNVs, indels, structural variants, and CNVs, and phase those variants to resolve haplotype information. In 2016, we launched our first single-cell product with focus on gene expression. The single-cell gene expression solution allows you to study the transcription profiles of millions of cells, identify clusters of cells with similar expression patterns, and isolate genes of interest based on their transcription profiles. This enables you to understand, among other things, the different types and quantities of cells that are present in your samples. Last year, we released our second single-cell product, which gives you the ability to interrogate the adaptive immune response and immune repertoire at single-cell level, again, across millions of cells. This comprehensive solution allows you to do things like discover clonally selected adaptive immune responses to different environmental and experimental conditions. And last week, we started shipping our newest product, the Single Cell Copy Number Variation Solution. This is the first scalable end-to-end product for profiling whole genome structure at a single cell resolution, and it will be the focus of our webinar today. So why is single cell genomics so important? This can be boiled down to the concept that different cells in your body have different genomes. As cells grow and evolve, mutations accumulate and propagate to create subpopulations of cells that have different genetic makeup than their progenitors. These differences contribute to the fact that cells have different types, lineages, and functions that determine things like tumor heterogeneity, neuronal mosaicism, cell line divergence, and the development of tissues and organs. Understanding single-cell genomics allows us to study these effects and characterize the clonal evolution that leads to uniqueness and diversity in both normal and disease states. The Single Cell CNV Solution provides a comprehensive, scalable approach to determine genomic heterogeneity and map clonal evolution by profiling hundreds to thousands of single cells in a single sample. With our Chromium controller and our single-cell DNA kit, which includes reagents and microfluidics chips, you can create sequence-ready libraries from the DNA of single cells and analyze the genomic DNA with our intuitive software and visualization tools. We have demonstrated our solution to work with a number of different sample types, including cell lines, primary cells, fresh and frozen tissue, and we are continually working to expand that list. Our technology generates uniform libraries across the genome, which allows us to call CNVs with a high degree of accuracy. We can accurately detect single-cell CNVs at a 2 megabase pair resolution and CNVs in groups of cells down to the resolution of a few hundred kb. The single-cell CNV solution gives you the ability to profile up to 5,000 cells in a run. Of course, you may conduct your own analysis on even more cells. And during Shimoni's part of the talk, she will discuss a custom analysis that we did to aggregate 5 sets of 2,000 cell data for a total of 10,000 cells in order to conduct the spatial characterization of tumor heterogeneity. With this unprecedented level of scale and resolution, it is now easier than ever for you to study complex pathogenesis, including cancer progression and genetic disorders. So this slide is an overview of the workflow, starting with the user-supplied cells. The library creation stage is a 2-step process that involves the creation of cell beads and then encapsulation of those cell beads with 10x barcoded gel beads to create a cell bead gel bead emulsion. I will go into a little bit more detail about this process in the next few slides. The resulting library constructs are then sequenced, and finally they are analyzed and visualized with our easy-to-use software. The single-cell CNV workflow is a 3-day workflow. This slide provides a high-level overview of the steps, and the first day consists of cell or nuclei preparation, followed by cell bead generation on the instrument. Day 2 consists of post-processing of cell beads, generation of the cell bead gel bead emulsions, and isothermal incubation to amplify the DNA. Day 3 involves the final cleanup and library preparation in order to then do the subsequent sequencing. So let's take a closer look at the library creation, which involves a 2-step partitioning process. The first step is the cell bead generation. This step, which is performed on a microfluidics chip, is run on the Chromium controller. As you can see in the diagram on the left, the cell bead polymer and cells, represented by the gray dots, are encapsulated into a cell bead emulsion, represented by the light blue bubbles with the gray dots inside. Each cell bead contains 1 cell. After collection of the cell beads, the cells are subjected to protein digestion and denaturation to clean up the DNA, which is still retained inside the cell bead. The second step is the cell bead gel bead generation. This step is performed on a second microfluidics chip and takes place on the Chromium controller. In this step, shown on the diagram on the right, cells are lysed and the cell beads containing the DNA are partitioned with 10x barcoded gel beads on a second microfluidics chip. This results in cell bead gel bead emulsion, also known as CBGBs. Each CBGB contains 1 10x barcoded gel bead and 1 cell bead, which in turn contains DNA from a single cell. This is a video showing the loading of the cell beads and gel beads into an emulsion and demonstrates our ability to accurately encapsulate single cell beads and single gel beads into an emulsion. The next phase is the DNA barcoding and library preparation phase. After the CBGBs are collected from the second partitioning step, the DNA is amplified and the 10x barcodes are added to that amplified DNA. Since DNA from a single cell is barcoded within a CBGB partition, DNA fragments originating from the same cell will share a common 10x barcode. Barcoded fragments are then pooled and attached to standard Illumina adapters. The 10x barcoded libraries are now ready for standard short-read sequencing to generate analysis-ready data. Up top, you can see the details of what the read constructs look like for paired-end sequencing. This final read construct can then be passed along for the sequencing. The Single Cell CNV solution is supported on the NextSeq, HiSeq 2500, 3000, 4000, and HiSeq X, and also the NovaSeq. To accurately detect 2 megabase pair CNVs at a single cell level for human samples, we recommend a sequencing coverage of 750,000 read pairs per cell, or 1.5 million reads per cell. Of course, trade-offs can be made between the detection accuracy, CNV resolution, and coverage. For example, if you need lower CNV resolution, you can do less coverage. You will see more information about lower coverage data presented by Shimoni during her part of the talk. And more details about these trade-offs and their implications can be found on our support site, which I will provide a link to at the end of this presentation. Once you have sequenced your libraries, you can use our software called Cell Ranger DNA to analyze the sequence data. The software is freely available on the 10x support site and easy to install. The Cell Ranger DNA software performs CNV calling and hierarchical clustering, and the key steps involve processing the reads and aligning them to the reference. Cell Ranger then identifies unique cells based on their barcode information. The reads are binned and normalized in preparation for CNV calling. Cell Ranger DNA then calls CNVs across the genome for each individual cell based on the binned reads. Finally, the single cell CNV profiles are then used to perform hierarchical cross-clustering. As a part of that process, aggregate CNV profiles are generated for groups of cells from that form a cluster. Using the aggregated read data from cells that have been grouped together by the hierarchical clustering process, we are able to get down to a few hundred KB resolution for CNV calls. Cell Ranger DNA outputs the results in standard file formats. Later in this webinar, Shimoni will talk about how you can use some of these standard output files to conduct a more custom analysis of the data. Cell Ranger DNA also generates a custom format called loop file that can be used to visualize the results in the Loop Single Cell DNA browser. The Loop Single Cell DNA browser is a visualization tool that provides an interactive view of the CNV profiles and clustering results. The loop file that is generated by the Cell Ranger DNA software can be uploaded into the Loop Single Cell DNA browser. With the Loop Single Cell DNA browser, you can view a heat map of the single cell CNV profiles, zoom into clusters of cells, search for regions of interest by genomic coordinates or gene names, and evaluate metrics for CNV calls on a per cluster or per cell basis. This allows you to assess your results in the context of other information such as CNV call confidence, cluster heterogeneity, gene annotations, and more. Now let's take a closer view at the data that is generated by the single cell CNV solution. In this experiment, we took one section of breast tumor and ran it through our single cell CNV solution. This resulted in the CNV profiles for 2,053 single cells. The image that you see on the left is the CNV calls aggregated across those 2,000 plus cells to generate a pseudo bulk profile. You can see that on average, this tumor sample has a ploidy of 3 with some deviations in both directions. Now let's take a look at the single cell data. The heat map on the right shows the CNV profiles across the whole genome for the 2,053 single cells. Each row represents a whole genome CNV profile for a single cell, and the colors reflect the copy number call that was given at a particular location in the genome. The tree structure on the left represents the hierarchical clustering of the CNV profiles, which allows us to see cells with similar CNV profiles closer to each other in the heat map. On the top, you can see a group of cells that correspond to the normal diploid cells. And on the bottom, you can see the cancer cells. You can also clearly see that not all of the cancer cells have the same level of amplifications in the same regions of the genome. For example, in chromosome 3, you can see that some of the tumor cells have a copy number of 3, while others have a copy number of 4. This heterogeneity is masked in the pseudobulk CNV profile because the copy number calls are aggregated across all the different ploidies of the tumor cells and also the diploid normal cells. The level of resolution provided by a single cell information allows you to understand the clonal structure of cancer and study the implications of that structure on disease progression, treatment responses, and drug resistance. So now that you have an overview of what the Single Cell CNV solution is and the type of information that you can generate, let's take a look at a demonstration of the kind of biological questions that can be studied with this solution. With that, I will hand the presentation over to Shimoni, who will discuss an experiment we ran to determine the spatial characteristics of genomic heterogeneity in tumors.
Thank you, Anushka. So I'm going to be talking about a specific use case of the Single Cell CNV solution, specifically to explore heterogeneity in cancer. So cancer is essentially a disease of uncontrolled cell division. You start with a normal cell that accumulates mutations, and through processes of drift and selection proliferates into a tumor. A tumor is effectively a heterogeneous mass of abnormal cells that express a range of phenotypes with differing evolutionary potential and fitness. This phenotypic heterogeneity or cell-to-cell variation has important clinical implications because cells within the same tumor can express different biomarkers. They can respond differently to treatment, or they may have different metastatic potential. So from a therapeutic point of view, understanding this diversity is critical. And a key driver of this phenotypic diversity is the underlying genetic heterogeneity. So we now know that tumors are frequently composed of multiple clones, each with their own genome. And specific clonal lineages expand or contract during tumor progression and in response to treatment. So the bulk genome of biopsies over time might look very different because the underlying cellular population has changed with clonal dynamics. And the cancer genome varies not only with time, but also in space. Clones may be spread unevenly throughout a patient's body upon metastasis, or they may even be spatially segregated within a tumor, as shown below. So to truly understand the genetic diversity in a tumor and to characterize the mutational spectrum, we need single cell resolution. And the original experiment to assay the genome at a single cell level was, of course, karyotyping. And what I'm showing in the left is an image, a spectral karyotype of a normal diploid cell that has 2 copies of each chromosome, each in its own color. You can contrast that with the image on the right, which is the single cell from a breast cancer cell line, which looks incredibly messy in comparison. And if you take a zoomed-in view of some of the marker chromosomes, you see that they're a mosaic of different colors, indicating massive genome-scale rearrangement where different pieces of different chromosomes have been stitched together. So in cancer, in addition to single nucleotide polymorphisms, there are large-scale genome rearrangements. And frequently, chromosome segregation errors occur in mitosis, commonly causing aneuploidies, where imbalance can range from extra or missing copies of whole chromosomes to varying degrees of copy number alterations and structural aberrations. And while karyotyping can beautifully articulate these complex structural mutations, it suffers from 2 major limitations. One is of throughput, and the second is of resolution. So we're going to be presenting Single Cell Genomics version 10x, where we have assayed spatial heterogeneity of a frozen breast tumor by sampling thousands of single cell genomes and assaying copy number variation at megabase pair scale resolution. So this is the major steps outlined below. So we began by harvesting nuclei from 5 sections of a frozen breast tumor, and the nuclei dissociation protocol optimized for single cell DNA is going to be available soon on our website as a demonstrated protocol. We then prepared libraries following the user guide for each section, sequenced, and analyzed that data using the Cell Ranger DNA pipeline, which aligns reads to the reference genome, normalizes for bias, and outputs integer-scaled ploidies for single cells. We then used the output of the pipeline to perform custom analyses in R using publicly available packages. We extracted a CNV event matrix from non-diploid cells, and we then clustered that. So let's get into the data. So we sequence the libraries in a NovaSeq using a paired N100 configuration, and for each of the 5 sections recovered approximately 2,000 cells. And for each library, we recovered around 2,000 million raw reads, which effectively translates to 600,000 to 900,000 mapped reads per single cell. This is very sparse coverage of the whole genome. It, on average, results in 0.02x coverage of the human genome in each single cell. And we're using the sparse genome coverage to call megabase pair scale CNV events. So another metric that helps in orienting is how many reads are present per megabase pair bin. And for this particular data set, it ranges from 200 to 300 reads. If you were to now aggregate all the data from single cells present in one section to get a pseudo bulk, we would have a depth of coverage ranging from 35x to 50x. So let's begin by looking at the pseudo bulk data. So on the left is an image of the tumor we analyzed, blown up, of course. And throughout this talk, the 5 sections are labeled A through E. And on the right are the pseudo bulk CNV profiles, which are effectively the average ploidy of all single cells in that section plotted in 2 megabase pair bins across the genome. And what's immediately striking is how different these CNV profiles look across the 5 sections. If you look at section A, it's a flat line at 2 with an average ploidy of 1.99. In contrast, if you look at section E, it has a much more interesting CNV profile showing multiple events and with an average ploidy of 3.39. If you now look at this data at single cell resolution, the reason for this becomes evident. So these are again heat maps. Anushka already introduced them, and they're very similar. So the columns are 2 megabase pair bins, the rows are single cells, and the colors indicate ploidy according to the scale. In addition, there's also this annotation bar which classifies the cells. So in green are diploid cells, in beige are non-diploid cells, and in dark brown are noisy cells. These are cells that are pipeline flags as having low confidence in the CNV profile. So it's quite obvious that section A is dominated by a diploid cells, and the diploid cells are dampening the signal from the small number of cancer cells, resulting in the pseudo bulk looking like a diploid. In contrast, section E has a majority of cancer cells. And so it results in the averaging, in the aggregate of the data, you can see CNV events. And so this just affirms what we already know, which is that tumor samples tend to be an uneven mixture of normal and cancer cells. And in addition, single cell resolution allows you to parse that mixture. Another way of looking at this data is by plotting the density profiles. So on the x-axis is the average single cell ploidy, and on the y-axis are your sections. So let's begin by looking at section A. You see a large peak centered at 2, and that's because most of the single cells in this section were diploid, so their average ploidies were in or around 2. In contrast, there's a much smaller peak between 3.5. You see the reverse for section E. Here you have a much higher peak centered at 3.5. These are your cancer cells, and a smaller peak centered at 2. So the 2 take-home messages from this plot are: 1, single cell resolution can cleanly parse this mixture of normal and cancer cells, and 2, it is now bioinformatically trivial to select the cells of interest, which in our case are the cancer cells, and remove from our analysis the contaminating normal cells. So in the next few analyses, I'm going to be focusing on this population of cancer cells across all 5 sections. So this analysis was performed in R using output files generated by the Cell Ranger DNA pipeline. So the goal here is to extract a binary CNV event matrix which tabulates the absence or presence of a CNV event across the cancer cells. So we begin with the bed file that includes within it genome coordinates of the CNV event, its ploidy, and which cell it came from. We selected events that were present in the 4,510 cancer cells that we identified from our previous analysis. We then filtered the events such that they were greater than or equal to 2 megabase pairs in size and had a confidence score of greater than 15. We then grouped events across single cells using two criteria. One, the event should have the same ploidy across the different cells, and two, there should be 90% reciprocal overlap of the events. Once we grouped the events, we could tabulate their absence of presence across the single cells. And finally, we discarded rare events that were present in less than 5% of the population. So after doing all this, we recovered 122 CNV events that ranged in size from 3 to 148 megabase pairs. And that data is shown in this matrix on the left. So this matrix has 122 columns for each CNV event, and it has 4,510 rows for each cancer single cell. Yellow indicates that a CNV event has been identified in that cell, and black indicates that it is absent. And what you can see is, here's a set of events that are present at a very high frequency. They seem to be present in nearly all the cells. And in contrast, you can also see events that are subclonal. For example, the ones here. These are present in a group of cells, but missing in others. So you can use these patterns of CNV as distinguishing features to cluster the cells. And this clustering can be done in many different ways. We decided to use discriminant PC analysis included in the AdGenet package. And what this does is it tries to maximize between group variation while minimizing within group variation. We arbitrarily chose to divide the data into 7 clusters. And those clustering solution has been annotated on the left of this matrix using the colors. And I hope it's clear to you that there's a shared pattern of CNV events which is being used to distinguish the different clusters. And next, we wanted to ask, well, how does the whole genome profile of these clustered cells look? We can see that they share events, but then how do they differ in the whole genome? And so that's shown in this heat map. While we're not showing the whole genome, we're focusing on a subset of chromosomes that have 3 or more distinguishing CNV events. And you can see how their CNV patterns differs from cluster to cluster. You can also pick up subtle trends such as chromosome 3 and 4 both show ploidies of 3 and 4. However, this pattern seems to be mutually exclusive. So this clustering and this heat map includes single cells across all 5 sections. So another question we can ask is, well, how are these clusters distributed across the tumor? And we're showing that in this stacked area plot on the left. So on the left is section A, which we know is composed primarily of diploid cells. And on the right is section E, which we know is composed primarily of cancer cells. But what's really interesting is that you see an expansion of clusters 1 and 2 in the middle of the tumor, and an expansion of clusters 6 and 7 on the other end. And if you look at the phylogenetic relationship between these clusters, you see that they are genetically divergent. So what we found is that in this tumor, genetically divergent clusters are spatially segregated. So in conclusion, we began with a tumor mass, which we deconstructed into its single cell constituent genomes, and by profiling single cell, the CNV profile of single cells, we were able to parse intermixed subclones. We also found that due to spatial segregation of subclones, a single biopsy of a tumor may be biased. And finally, we found that single cell genomics can simplify inferring cancer evolutionary paths. And with that, I will hand it back to Anushka.
All right, thank you, Shimoni. So as you've now seen, our single cell CNV solution provides a comprehensive, scalable approach for characterizing genomic heterogeneity and studying its impact on clonal evolution, disease progression, treatment response, and more. We have a number of resources available that provide additional information about this solution. There are videos from both poster presentations and workshop presentations available on our website. We have an application note that provides an overview of the solution with additional application demonstrations on the way. We also have publicly available data sets that you can download and play around with. Our support site contains a wealth of information, including our single cell CNV user guide, technical notes on the products, software documentation, and the downloads for the software executables themselves. And we will continue to update the support site with demonstrated protocols and other technical documentation as they become available. Finally, please feel free to visit the product page on our website to get more information about the Single Cell CNV solution, including product specs, and also to request pricing information. And with that, I would like to thank you for your attention. And now I believe we have our Q&A.
Thank you, Anushka and Shimoni, for a great presentation. Before we get started with our Q&A session, I'd like to introduce 10X R&D scientist Claudia Catalanati and 10X support scientist Laura Demare. They're here to help answer your wet lab questions today. Please remember to select the Q&A box on the bottom of the screen to submit your questions, and we'll do our best to get through as many questions as possible. All right, let's get started. And here's the first question. Have you seen any issues with generation of the cell beads when using highly heterogeneous cell samples, specifically where there are a lot of different cell sizes and maybe morphologies?
Okay, so in terms of cell recovery, no, we haven't really observed much of a difference. We do have a limited in cell size, so as long cells are within the same range, we don't really see a decrease. Obviously, cells that are larger than 30 micron, larger than 40 micron, you have a risk, or you rather have a risk of increased risk of clog, but we haven't really seen much of a diversity due to the type of sample that we have analyzed.
Okay, thank you. Next question. Can you explain why there's a separate cell bead, which is different from the single cell RNA-seq protocol?
Yes, so the cell bead allowed you to treat separately the cells, to lyse the cells, to remove protein and denature the DNA. Those are all pretty harsh treatments, and that's why it has to be separated by the usual gem in the single cell 3', allow you to access more easily to the DNA.
All right, and next question. Some germline CNVs do exist. Is it necessary to do a tumor normal comparison?
So I think that depends on what the proportion of that mutation is in the sample. So if you saw that even in the breast tumor, you also sample normal cells are present in that. So within that sample, you might be able to access some of the normal cells and then subtract out the mutations present with the ones you see in your tumor sample.
Yeah, I think just to add to what Shimoni said, the fact that you are able to identify within a tumor sample both the tumor population and the normal population allows you to basically construct your analysis however you want, whether you want to actually analyze just the normal cells, just the tumor cells, or do a comparison. That's entirely up to you.
All right, next question. How many cells do you need to aggregate to be able to call single nucleotide somatic mutations?
Yeah, that's a great question. So most of our effort on the analysis side of things has been focused on CNV calling. We are continuing to look into what the level of resolution and accuracy is for SNV calling. So while that's not officially supported as part of the solution, we do know that some of our early customers are already starting to do SNV analysis on the data. At this point, we would say on the order of hundreds of cells at least would need to be aggregated to do SNV calling, but it really depends on the heterogeneity of this sample and how homogeneous the clusters of cells are that you're basically aggregating across.
And if I can add, depending on what the copy number profile is, if you have an SNV that's present in a homozygous, in a deletion or one copy of the genome, it's much easier to pick it out versus an SNV that's present on one haplotype, which is present in a higher copy number.
Great, thank you. Next question. What is the range of the confidence score? How is it calculated?
Yeah, that's another great question. So we definitely encourage folks to take a look at the support site, which has a lot more detail about the software, underlying software algorithms. The answer to that particular question is based on how we actually do the CNV calling. The confidence scores essentially it represents how confident we are in that CNV call relative to the next best solution. And the range of the confidence scores goes from 0 all the way up to 250, but really, or 255, but really anything above a confidence score of 2 is considered a good, a high confidence score.
Right. Next question, or this is really more just asking about people's thoughts. In this case, it appears the tumor grew from one end to the other. Is this a common finding, or do tumors grow more organically, unpredictably? I don't know if anyone has experience with this.
My guess is really how you get the biopsy tissue. So that's my best guess.
All right, thank you. Next question, how many million reads per cell did you say you recommend?
Yeah, so the recommended coverage to get 2 megabase pair CNV event resolution at a high confidence or highly accurate calls is 750,000 read pairs per cell, or 1.5 million reads per cell. And you can lower that coverage depending on how you want to make the trade-off of the accuracy of the CNV calls or the size of the CNV events that you're interested in. If you're interested in larger events, you don't need to have as higher coverage. You can drop the coverage if necessary. And we have information about the trade-offs between the size of the events that you want to detect and the implications of the accuracy on the lower coverage, all available on the support site.
All right, next question. How have you dissolved the tissue into single cells?
So we are going to have soon a demonstrative protocol on how to isolate nuclei from frozen tissue. So we actually we cut the tissue in small pieces and we isolate from each section nuclei and run the experiment. The protocol is coming up soon on the website.
All right, thank you. Next question. Can you comment on the quality of the data with lower coverage, meaning reads per cell? What is the minimum reads per cell?
Yeah, I think, so for the CNV calls, again, I would recommend looking at the information on the website. We basically have provided some curves that demonstrate as you lower the coverage, what the implications are on the accuracy of CNV calling compared against a truth set that we were testing against internally. The minimum reads per cell really depends on the biology that you're trying to do. If you're trying to detect, chromosome scale events, you're going to need much less coverage than if you're trying to, again, detect smaller resolution events.
Yeah, and I'll say to Anushka's point, it's about the biology. So if your cell is diploid and you're only expecting changes that are going up to, say, tetraploid, then you may not need that much. Versus if you're seeing, a cell that is, octaploid and you want to see smaller gradations of changes, that makes things more complicated.
Right, thank you. Next question. Have you ever encountered problems or differences in lysing cells from different samples, for example, tumor entities or species?
So in terms of cell lines, no, in terms of lysis, we have, again, on our dissociated protocol, we have tested several types of tumor types. You might need to optimize, you might need to adjust a little bit the lysis time or the way you break the cells, mechanically break the cells. But for the tissue type that we have tried, we didn't have problems in efficiency. We tried a couple, we tried breast, lung, very early on, other tissue type.
All right, next question. I noticed in the breast cancer sample that the Y chromosome was not shown as copy number 0. Was the data processed such that it wouldn't show? And if so, does this affect the ploidy calls for the other chromosomes?
It actually is. It's such a small part of the genome. Do you see that I'm pointing my arrow at it? And it's dark, it's that dark blue. So it is shown as 0.
All right, thank you. Next question. Have you tried single nuclei instead of single cells as input, and how's the performance?
Yes, actually the entire presentation is all done on single nuclei from tissue.
Frozen.
Frozen, yeah, frozen tissue.
demonstrate?
The demonstrated protocol is coming along on the website soon.
And that'll be for nuclei.
Yeah.
Great, thank you. Next question. If interested in the epigenetic marks in the DNA, can the amplification step of the DNA in the beads be skipped and recover the unamplified adapted molecules?
Yeah, I have to say, I don't think we're sure about. We definitely have not looked into that, so I don't think we have a good answer for that. I would say, if you want additional questions, maybe email support, and we could see what or support at 10xgenomics.com, and we can see what other information there might be available.
All right, thank you. Next question. For frozen nuclei extraction, what is the smallest mass size you start with in order to obtain enough number of nuclei for CNV?
So again, in our demonstrated protocol, we have tried a range of size, and we normally recommend between 2 to 4 millimeters, cubic millimeters, and that will give you enough materials to get enough nuclei to go through the protocol.
Okay, thank you. Next question. How is the DNA sampled from the single cells? Degenerate oligonucleotides? In other words, how are the reads themselves generated from the genomic DNA?
So the DNA is amplified using random hexamers during an isothermal incubation step. And then after the amplification, the DNA is barcoded with the 10x barcode. And that allows you to identify which cell the amplified DNA came from.
All right, thank you. Next question. Have you tried combining the single cell DNA with the single cell RNA from the same samples, or can they be complementary to each other?
So we have done it on the same sample, but not the same cell. So you can take the same sample, split it up, run the single cell gene expression protocol, and run the single cell DNA protocol. And not going to say more, but they looked very interesting and quite complementary to each other.
All right, thank you. Next question. What is the cost of the kit and how many samples can be run simultaneously?
So in terms of simultaneous samples, you can do 8 samples in parallel on the chips that are provided. For pricing information, I would recommend to either contact your sales representative, or you can go to the product page and request a quote.
Thank you. Next question. How do you define the ploidy? Do you have some suggestions on how to distinguish doublets and high ploidy cells?
So the way that we identify the copy number calls is based on the number of reads that are present in a particular bin size. So if you recall, we do the alignment of the reads to the reference genome, and then we partition the reference genome into bins. So depending on how many bins are in, how many reads are in a particular bin, looking across the genome, we do some normalization and then look at the relative comparison of the number of reads across the bins, and that's how we identify the copy number calls. And then, sorry, there was a second part to that question. We don't. Yeah, I think I don't have an answer to the doublet question off the top of my head.
So if you wanted to bioinformatically analyze doublets, it's hard. I mean, of course, if you have 2 different species, you can figure that out. The other way that we have identified doublets is if you have a larger, well-known CNV event in one cell type, which is absent in the others, then when you have a doublet, you'll start seeing patterns where that CNV event is present at half the ploidy value. So that's a bioinformatic trick that can be used, but then you do need to understand already the profile of the cells.
And I'll also add that as far as the expected multiplet rate when running single cell CNV, it's comparable to our single cell 3' and BDJ solution. So the multiplet rate is quite low. It's 0.7% for every 1,000 cells you target.
Great, thank you. Next question. Are you thinking about doing single cell CNV on non-human cells, like Plasmodium with smaller genomes?
Yeah, so we right now have done all of our testing and sort of quantification and validation of accuracy on human samples. We have done a number of experiments on mouse, but that's all that we've done in-house. There's nothing that says that this solution can't be used, and we do encourage people to definitely try other organisms. Unfortunately, at this time, we don't have any good guidance or recommendation.
And the pipeline, the analysis pipeline, is agnostic of the species.
That's true, yeah. For the analysis pipeline, you can actually specify your own reference sequence, and so you can run it on something that is not non-human.
All right, thank you. Next question. Do you have to use a separate cell range or DNA to call the CNVs?
So once you do the sequencing and you generate the output, the sequence data, that gets input directly into the cell range of DNA pipeline. That is the primary analysis pipeline that takes the raw sequence data and generates the CNV call. So if that question, I'm not sure if that question is coming from familiarity with other products where we have Cell Ranger for our gene expression product. The Cell Ranger DNA software is a separate software that is specific for our single cell CNV solution. So I hope that answers your question.
All right, thank you. Next question. How can we eliminate or decrease the noise cells?
The noisy cells flagged by the pipeline. So that's an interesting question. And so a little bit more detail on what noisy means. So it's actually taking into account two parts. One is what the confidence is in the integer scaling, right? And then the other is you look at the CNV profiles within that sample and say, okay, does this cell, is its CNV profile an outlier? So often if you have, say, a sample or a cell line that's undergoing replication and it has a lot of up and down changes in its CNV events, that can get flagged as noisy. So it's just a guideline for you to, look at the data and highlight and say, okay, I should be looking at these cells a little bit more closely. It doesn't necessarily mean that there is a problem with the cells.
All right, thank you. Next question. Is there any QC included in the process?
So on the workflow, on the library preparation, there is similarly to other protocol from 10x. You have the post-gem generation. Once you break the gem, post-isothermal amplification, you will have a bioanalyzer that will tell you how much DNA you actually have managed to barcode. And then you will have a final bioanalyzer trace once the library is completely converted, ready to go on the sequencer, and then qPCR to quant the library.
And then on the analysis side, one of the outputs of the Cell Ranger DNA pipeline is a web summary that provides a lot of information and metrics to evaluate the number of cells that were called, the types of CNV events, and the ploidy, and a lot of other types of metrics that can be used to QC and evaluate the results.
All right, next question. This is a follow-up to a previous question that was answered regarding the ELYSIS efficiency. And you just the asker is clarifying, this question was regarding the lysing of the cell within the system, and if you'd seen any differences with different cell types or species. And if you could clarify, that'd be great. Thank you.
Okay, so different species we haven't tried. In terms of different cells, different tumor type, we gained generally the same library complexity per samples. If that is the answer to the question.
All right, thank you. Next question. You recover around 15% of your input. Do you lose most of your cells in chip 1 or 2?
So there is something that is in the protocol. So you record 85% of your cells get incorporated into cell beads in step 1. You have between the chip 1 and the second chip, you are treating your cells and then you are filtering again your cells. So the loss is even due because you are leaving behind some cells between the process. So I would say there is the same loss across the 2 chips. It's just there are extra treatments before you go into germ.
All right, thank you. Next question. Have you tried nuclei from FFPE samples?
Not at the moment, and it's not supported.
All right, next question. Chromosome 3 and 4 pattern from the single cell DNA is very interesting. Do you see the same pattern from bulk sequencing?
Okay, so you're referring to this pattern, right? So we can go back and look at the pseudobulk profile. And no, so actually what happens is, because of averaging across all the cells, which are also including the normal cells, you actually get a flat line of 3 and 4 in the bulk across all 5 sections. So no, unfortunately, when you do bulk, you miss that interesting signal. Thank you for the question.
All right, thank you. Next question. Is it possible to modify the protocol to look only at targeted genomic regions and increase coverage?
So the workflow itself, you can modify and you can actually do targeted. That said, is not an analysis that we support at the moment. So I don't know if someone from the analysis wants to.
Yeah, so it's not something that we have tested in the analysis software, but the workflow is compatible with doing target capture.
Great, thank you. Next question. What is the expected run-to-run variation in percentage?
Percentage what?
Yeah.
Well, if the person who asked the question is on, if they could clarify, I'll go to the next question and come back to that one. So I'll move on to the next one. Is it a good idea to aggregate the CNVs for all cells?
I'm going to interpret this question. So there are 2 ways of aggregating. You can aggregate across the same bin. So that would give you the pseudo bulk profile. The other thing you can do is you can aggregate all the bins of the same cell, and that'll give you the average ploidy of the single cell. So they give you 2 different kinds of information.
So as part of the pipeline, we will output both individual CNV calls from individual cells, and as part of the hierarchical clustering process, every time cells are grouped into or aggregated into a group, we will do aggregated CNV calling. So you will get that information for both the single cell level and all of the aggregated groups throughout the hierarchical clustering. And then determining on depending on the heterogeneity of your sample really depends on, will really impact whether or not that aggregated data is meaningful across multiple cells.
All right, thank you. And we just have one question left. And so if you have any more questions, I urge you to put them in the Q&A box at this time. And here's the last question we have so far. How do you ensure that the absence of evidence of a CNV is evidence of absence?
You mean in the sense that there might have been reads that didn't get sequenced or regions of the genome that didn't get sequenced? I'm not sure I understand the question. Yeah, I think that one probably warrants a longer discussion.
Yeah, a longer discussion. A little bit of. All right, here's another question that came in. Oh, so here's a clarification for the question that was asked about the run-to-run variation. How much variation would you expect between runs from sample preparation to the end of the process?
Okay, so the way again I see this question, we have run same samples multiple times across different users, and there is a perfectly overlap and similarity and reproducibility of the results.
All right, thank you. Next question, what is the range of fraction of mapped reads? Would 45% be high, and if so, what may cause it?
So I actually don't know the answer to that off the top of my head, but I think that's something that, do you have an answer for somewhere?
I guess they require more specification. The range of fraction of mapped reads to the genome is actually higher.
It's much higher.
It's much higher. So
yeah.
if the person can actually clarify the question, because it is meaning the range of how many fractions are mapped to the genome, that is much higher than 45%.
Yeah.
All right, thank you. And that was our last question. And so that about wraps it up for today. Thank you to our speakers, Anushka, Shimoni, our 10X scientists, Claudia and Laura, and especially to our attendees for some great questions. We'll be posting a recording of this session on the 10X website shortly. If we didn't get to your question, or if you have a follow-up question, we urge you to post it on the 10X community in our single cell forum, or you can also email support at 10xgenomics.com. And finally, we have 3 more single cell webinars scheduled in the upcoming weeks. We invite you to join us to learn more about our single cell solutions for ATAC-seq, mouse immune profiling, and feature barcoding for simultaneous detection of gene and protein expression, or gene expression from CRISPR-mediated perturbations in the same cell. You can find out more on www.10xgenomics.com/celebrate-webinars. Thank you and have a great rest of your day.